She was staring at a spreadsheet that was rewriting history. Not the kind of history that sits quietly in textbooks and collects dust on university shelves. The kind that reaches back 12,000 years and pulls apart everything a civilization has ever believed about itself. Margaret Antonio, a researcher working with Jonathan Pritchard's genetics laboratory at Stanford University, had spent months extracting, sequencing, and analyzing ancient DNA pulled from the bones of 127 people buried in and around Rome. People who had lived, breathed, loved, and died in the city that built the Western world. And what the genomes were telling her, in the language of nucleotides and alleles and ancestral population models, was something that no Roman emperor, no Latin historian, no Italian nationalist of any era had ever suspected. The people who built the Roman Empire were not, genetically speaking, primarily European. At the height of Imperial Rome, the period between 27 BCE and 300 CE, when the city sat at the center of an empire of 50 to 90 million people stretching from the Scottish Highlands to the deserts of Mesopotamia, fewer than 30% of Rome's inhabitants had genetic ties to the early Italian ancestors who had lived in the peninsula before Rome's rise. The rest, the majority, the genetic plurality of the greatest city in the ancient world, traced their ancestry primarily to the eastern Mediterranean, to the Near East, to Anatolia, and to North Africa. The DNA of ancient Romans looked less like the DNA of modern northern Europeans, and more like the DNA of people from what is today Syria, Lebanon, Turkey, and Egypt. The city that gave the world Latin, Roman law, the Catholic Church, and the architectural template for every government building on Earth was, at its peak, genetically a city of the East. And almost nobody knew. The study that revealed this was published in November 2019 in the journal Science, one of the two most prestigious scientific publications in the world. Its lead author was Margaret Antonio. Its senior authors included Jonathan Pritchard, a professor of genetics and biology at Stanford University, Alfredo Coppa, a professor of physical anthropology at Sapienza University of Rome, and Ron Pinhasi, an associate professor of evolutionary anthropology at the University of Vienna. Together, these researchers gathered 127 ancient DNA samples from 29 archaeological sites in and around Rome, with dates spanning from approximately 10,000 BCE to the medieval period, a window of roughly 12,000 years of human presence in a single location. To extract usable DNA from remains that old, the team used a technique that has only become reliable in the last decade or so. They drilled into the cochlear portion of the petrous bone, the densest bone in the human skull, located behind the ear, where ancient DNA is best preserved against the degradation of time. They sequenced the genomes to a median depth that allowed robust analysis, and they ran the resulting data through multiple established statistical frameworks for ancestry modeling, comparing each ancient individual's genetic signature against a vast reference database of both modern and ancient populations from across Europe, the Middle East, North Africa, and Central Asia. What they found was not a single revelation, but a sequence of them, a layered story of population change so dramatic, so repeated, and so complete that by the time the paper was finished, the researchers had effectively produced the first genetic history of Rome across 12 millennia. Every major historical turning point that Latin historians had recorded in words, the arrival of farmers, the Bronze Age migrations, the rise of the city, the expansion of the empire, the fall of the West, the rise of the medieval, showed up independently and unmistakably in the DNA of the dead. The story begins, as all Italian stories ultimately do, with people who left no written record. 12,000 years ago, in the years immediately following the last ice age, the Italian peninsula was home to small, scattered groups of hunter-gatherers. The three oldest genomes in the study, dating to between 9,000 and 12,000 years ago, belonged to people whose DNA closely resembled other Western European hunter-gatherers of the same period. These were the original inhabitants, the first Italians in any meaningful sense, and their genetic signature became the baseline against which everything that followed would be measured. The first great transformation came approximately 9,000 years ago, and it was not subtle. Farming arrived in the Italian peninsula, or more precisely, farmers arrived. The people who brought agriculture to Italy were not the hunter-gatherers who had been there for millennia, gradually learning new skills. They were a distinct population, arriving by sea and by land from Anatolia, the region that is now Turkey, bringing with them not just domesticated crops and animals, but an entirely different genome. These Anatolian farmers were so genetically distinct from the existing hunter-gatherers that their arrival shows up in the ancient DNA record not as a gradual blending, but as a near replacement. Within a few generations in the genetic record, the hunter-gatherer signal drops sharply, and the Anatolian farmer signal surges. The first wave of deep ancestry shift in Italian history was not a meeting of cultures. It was a demographic transformation, This finding is consistent with what ancient DNA research across Europe has shown more broadly, that the spread of farming across the continent was driven primarily by the movement of actual people from the Near East, not by the transmission of ideas between existing populations. But Italy's position at the crossroads of the Mediterranean made it one of the key entry points for this transformation, and the genomes from Rome capture it with unusual clarity. The second great transformation came in the Bronze Age, roughly 5,000 to 3,000 years ago. Into Europe, from the east, from the Pontic-Caspian steppe, north of the Black Sea and Caspian Sea, came a population known to archaeologists and geneticists as the Yamnia, or their descendants, sometimes called corded ware people. These were semi-nomadic pastoralists, herders of cattle and horses, speakers of what linguists believe were early Indo-European languages. Their genetic signature, sometimes called steppe ancestry, swept across Europe with extraordinary speed in the genetic record, reaching Scandinavia, Central Europe, and eventually the Italian peninsula. The Stanford study confirmed that this steppe component arrived in northern Italy by at least 2000 BCE and in central Italy by 1600 BCE. The second wave of genetic transformation is the one that brought to Italy the ancestral population associated with the emergence of the Italic languages, the Latin that Virgil wrote in, the Oscan that competed with it, the Umbrian of the Central Highlands. By the founding of Rome, traditionally dated to 753 BCE, the genetic composition of the region had been shaped by all three of these layers: the original hunter-gatherers, the Anatolian farmers who had largely replaced them, and the steppe ancestry Bronze Age migrants who had in turn reshaped the farmer populations. By the Iron Age, Antonio and her colleagues found the population of central Italy genetically approximated what modern Mediterranean populations look like today. The Roman people, at the moment of Rome's founding, were in that sense already a complex, layered, deeply mixed population. And then the empire began. This is where the study delivers its most startling finding, the one that made it international news, the one that rewrites the popular image of ancient Rome in ways that historians will be absorbing for years to come. As Rome grew from a city-state into a republic and then into an empire, it became the single most connected metropolis the ancient world had ever produced. At its peak, the city of Rome had a population estimated at between 1 and 2 million people, a scale of urban density that would not be seen again in Western Europe until the 18th century CE. People moved to Rome from across the empire. Soldiers completing their service, traders seeking wealth, scholars and artisans attracted by patronage, enslaved people transported by force from every conquered territory. The geopolitical reach of Rome was effectively its immigration policy. And its immigration policy was, it turns out, its genetic policy as well. The ancient DNA makes this visible with a precision that no historical text ever could. During the imperial period, between 27 BCE and 300 CE, the genetic profile of individuals buried in Rome shifted dramatically and decisively away from the European and Mediterranean baseline that had characterized the population at the city's founding. The dominant ancestry signal in imperial Rome was Near Eastern, specifically Anatolian, from what is now Turkey and the broader Eastern Mediterranean. Contemporary Roman historians had noticed this in their own way. They wrote about the influx of people they called Greeks, a loose term, that reflected the fact that Greek was the dominant language of the Eastern Mediterranean world, and that many of the newcomers, whether they were from Anatolia, Syria, the Levant, or Egypt, arrived speaking Greek rather than Latin. The genetic evidence now confirms what those ancient writers were observing. At the height of Rome's power, when the Forum was at its most crowded and the Colosseum was freshly built, and the legions were garrisoned from Hadrian's Wall to the Euphrates, the people walking the streets of the city itself were predominantly of Eastern Mediterranean descent. The enslaved population, conservatively estimated at somewhere between 10 and 30% of the city's total population at various periods, was drawn from across the empire, with significant numbers from the Near East, North Africa, and Greece. Traders and merchants from Alexandria, Antioch, and Carthage maintained permanent communities in Rome. Soldiers who had served in the Eastern legions retired to the capital. Freedmen and freedwomen who had won or purchased their freedom remained in the city where they had worked. As Christina Kilgrove, a Roman bioarchaeologist at the University of North Carolina at Chapel Hill, who was not involved in the study, told Science magazine, the city's growth from a small town into an important city brought probably a lot of migration happening, an assessment that the genomes confirm with a thoroughness that even she found remarkable. Jonathan Pritchard, one of the paper's senior authors, framed the discovery in terms that cut directly to its importance. The historical and archaeological records tell us a great deal about political history and contacts of different kinds with different places. Trade and slavery, for example, he said. But those records provide limited information about the genetic makeup of the population. What the DNA provides, in other words, is the thing that the written record cannot: the actual biological identity of the people, beneath the Latin names they were given, beneath the Roman citizenship they may have held, beneath the toga that was the uniform of a civilization built on the fiction of a unified Roman people. The story that Rome told about itself, the story of descent from Romulus and Remus, of an Italian people who conquered the world through virtue, discipline, and martial excellence, was a political narrative. The genome does not care about political narratives. And the genome says that at the moment when that narrative was most powerfully asserted, when Roman identity was most vigorously policed through law and language and religion, the actual bodies of the people asserting it were predominantly descended from populations in modern-day Turkey, Syria, Lebanon, and the broader Eastern Mediterranean. This is not a diminishment of Rome. It is, if anything, a remarkable confirmation of something Rome itself claimed to believe, that Roman identity was a matter of law and culture, not blood. The great Roman orator and statesman Cicero, writing in the 1st century BCE, argued that Rome's greatness derived from its ability to absorb and integrate peoples from across the known world. The DNA suggests that this absorption was not merely rhetorical. It was literal, biological, and demographically dominant. But the story does not end with the empire's peak. The genomes from the study trace what happened next, and the pattern continues to surprise. After 300 CE, as the Western Roman Empire began its slow fragmentation under pressure from internal instability and external migration, the genetic signal in Rome shifted again. This time, the shift was toward European ancestry, specifically from populations associated with the Germanic and Gothic groups that moved into Italy during the migration period, sometimes called, with varying degrees of accuracy, the barbarian invasions. The Visigoths, the Vandals, the Ostrogoths. These groups left their mark not just in the historical record of sacked cities and deposed emperors, but in the DNA of the people buried in Roman soil during and after this period. A clear increase in Central and Northern European ancestry appears in the genomes from late antiquity and the early medieval period. The Near Eastern ancestry component that had dominated imperial Rome recedes, and a more European signal gradually reasserts itself. This means that the modern Italian population, and specifically the genetic profile of modern Italians that current ancestry testing companies use to define what Italian DNA looks like, is not primarily the DNA of Julius Caesar's Rome, or Augustus's Rome, or Hadrian's Rome. It is the DNA of a population that was itself the product of multiple subsequent waves of migration, recomposition, and replacement, built on top of the already extraordinarily complex genetic legacy of the empire. A separate study published in Science Advances examining the population structure of modern-day Italians using 2.5 million single nucleotide polymorphisms across more than 300 individuals confirmed this complexity from the modern end. It found that Italians today represent the largest degree of genetic population structure detected anywhere in continental Europe, meaning that within the Italian peninsula, there is more genetic variation between different regional populations than exists between many pairs of nationally distinct European countries. A person from Calabria and a person from Lombardy may be genetically more different from each other than a Spaniard is from a Portuguese person. That internal diversity is itself a product of the layered history the ancient DNA reveals. Different regions of Italy absorbed different combinations of the same waves of migration: Anatolian farmers, steppe pastoralists, Near Eastern imperial migrants, Northern European post-Roman arrivals, Greek colonists in the south, North African influences in Sicily, in different proportions and at different times. The Italy that exists today is not a single genetic population. It is a palimpsest, a document written and rewritten across 12,000 years of human movement, with every era's inhabitants leaving their mark in the bodies of the people who came after. The study also touched on something that has become one of the most philosophically interesting findings to emerge from ancient DNA research more broadly. The ancient Romans, whose genomes were sequenced, the people buried in Rome during the height of the empire, whose Near Eastern ancestry has now been confirmed by the most rigorous genetic analysis available, would not have thought of themselves as anything other than Roman. They spoke Latin. They observed Roman religious practices, or the new Christian practices that were spreading through the city from the east. They held Roman citizenship, operated within Roman law, competed for Roman honor. The genetic signal that marks them as predominantly of Near Eastern descent was invisible to them and to everyone around them. As researchers involved in the broader field of ancient DNA have noted, the people who moved to Rome from Anatolia and the Eastern Mediterranean almost certainly saw themselves as citizens or subjects of Rome, not as members of a distinct foreign ethnic group. The identity of Rome was, in a very real sense, larger than the biology of the people who held it. Rome absorbed the world and called it Roman, and the world, for the most part, agreed. What the Stanford study ultimately gave us is something more disruptive than a simple revelation about ancestry. It gave us a genetic mirror held up to one of history's most powerful and enduring myths, the myth of a coherent, continuous, biologically unified people tracing their descent in an unbroken line from a storied ancient past. That myth has been told not just about Rome, but about almost every major civilization. And ancient DNA, again and again, tells the same story in response. There's no unbroken line. There are only waves. Every population we study, when we look closely enough at its bones, turns out to be the product of migrations its later descendants did not know had happened, of people who arrived from places that later histories forgot or denied, of genetic contributions from groups that the founding myths of the civilization excluded or erased. The modern Italian is the descendant of hunter-gatherers, Anatolian farmers, Eurasian steppe herders, Eastern Mediterranean imperial migrants, Northern European post-Roman arrivals, and every combination of the populations that moved through the Italian peninsula in the 12,000 years since the last ice age. There is no single ancestry hidden in Italian DNA, because there is no single ancestry. There are layers, each one built on the last, each one a consequence of movement and mixing that the people doing the moving and mixing mostly did not plan or intend or even recognize. The 127 genomes extracted from 29 burial sites in and around Rome did not just reveal who the Romans were, they revealed something about who everyone is. That identity is a story we tell about ourselves, and ancestry is the much longer, much stranger, much more surprising story that the bones tell when nobody's listening.